So I'll just go ahead and start. The title of this presentation is kind of long and dry, but it's, we want to explore what automatic interoperability might mean for doing your scientific research. So trying to motivate this, but I think a better title would be just, well, what if a lot of data were already fair? What would that mean? What kind of capability would that give us? And I'm just going to start with kind of noting some very popular cases that are just kind of like in the popular press. I've seen these. Maybe many of you here are aware of even better examples of this kind of thing. But it's the idea of patients more and more having an interest in their own medical data. And certainly a patient is motivated like no one else to try and come up with ideas about how they're going to deal with a condition that they might have. And let me turn off. Do not disturb. So this is just the idea that, patients have access in some cases to their data like never before. They certainly have access to online information like never before, and maybe that's partly a good thing, maybe it's partly a bad thing. But you're seeing more and more examples like this. So this is a TED Talk. Dave wants to help patients help themselves by owning their own medical data, connecting to fellow patients, and by making medical care better. So the idea is implicit here that by connecting data, you can only expect better outcomes for kind of navigating your condition. There's another interesting example from this guy, Jack Adraka. He was a high school student in a biology class, sort of at that level of education. I think his brother had pancreatic cancer, and so he became very interested in this and just went online. And it was essentially Google and Wikipedia that allowed him to create a new diagnostic for pancreatic cancer that was very well received. I think there's been some controversy lately, but in essence, Jack drove science further because he had a good idea, and even if people are testing it now, it's still perfectly good science. So this is an example where even a non-specialist, right, maybe has the latitude to operate in this data space that a professional might not, just because of the demands of everyday work in a professional environment. So, but that data is out there. If amateurs can access it and piece it together, why can't computers? Right, that's kind of the message, I think, from Jack. And then this is an article from The Atlantic, and this is Larry Smarr, and he's, I think, known really as a kind of a tech entrepreneur in Silicon Valley. Has training in mathematics and physics, but he has, among other things, Crohn's disease. And being the analytical scientist that he is, he's now collecting data on like everything of himself. When I saw him speak before, he was doing like time-delimited metagenomic sampling on his own gut microbiota. But he's figuring out ways to record everything he can about himself. So it's a real like example of self-quantification. But the idea here is that it's the future of healthcare, as far as he sees it. And so these are patients, these are data sets, and it's kind of an effort to bring all this together. In our group, in Leiden, in GO-FAIR, we've run across an individual who's also kind of a tech-savvy person. His initials are JVDB. And, but here's a real case study that we encountered just in the last year. And the way this goes is that this patient had gotten thyroid cancer, and his thyroid was eventually removed. And about 2 years later, he ended up developing COPD. And so his question to himself, for various reasons, was, is there a causal link between the removal of my thyroid and the occurrence of COPD? I don't think he had a history of heart condition in his family, so it was kind of unusual. He consulted his doctors. He went to his physicians and asked, is there a link? And the physicians came back negative. No, there's no link. He then went to PubMed, and he also found kind of negative results. In general, it's likely that his physicians were probably just doing searches on PubMed as well. So in any case, there were negative results there. He ended up kind of launching himself onto this medical odyssey after that, where he went to all these different hospitals that he had visited and all these different clinics, and he tried to piece together or to accumulate all of his own personal medical records and data. And it took 2 years. I don't think it was always easy for him to do that, but he gathered his patient records. There's an organization called Adraka in the Netherlands that is contracted to handle medical records. He ended up, I guess, getting like 50 terabytes of data in the end because he had some scans done as well. He was reading the literature and doing advanced literature searches. And at the end of all this, he thinks he has identified 3 candidate proteins that might actually link thyroid glands and COPD. And this CXL12 was like downregulated, and that caught his attention for various reasons. So his question to himself was, okay, perhaps if I can regulate the level of the protein of CXL12, maybe I can remediate this COPD. He also went on another app, a health tap. This is an app where you, I think, cash in $99, and then you can send your questions to physicians. And physicians who see your question and have some feeling of authority there can respond to your questions. So he asked, is there a relationship between thyroid gland and COPD? The physicians were saying, no, there's no connection. However, one of these physicians said, no, but I'm wondering why you ask this question, because I see this question all the time. Or I've seen it more than once anyways. The patient said, yeah, and the doctor said that, the physician said, this is an interesting question. I see it multiple times. Can I call you? And that's when the patient said, yeah, you can for $99. So he's kind of reversing the health tap business model. But then eventually the patient got in touch with this company called Eriks. We've worked with them in Leiden, but this is a company that has done kind of in a manual way the integration of like 200 different life science data sets. And within minutes, he could run a query asking, is there a relationship between COPD and thyroid gland, and what are those relationships? And 5 proteins came back, 3 of them the ones that he had already been looking at. So it's an example of where the computer is starting to assist someone in a few minutes in what otherwise took 2 years of work. So given that use case, right, so we involved like PubMed, Adraka, HealthTap, and Eriks. So 4 different data sources. I'm going to break out of here, and I'm going to show a short 2-minute video.
The Personal Health Train's main goal is connecting health data to make them more usable. Personal health management requires increasing amounts of personal data, but today this information cannot easily be used because the data are produced and managed by various healthcare providers, authorities, and more by citizens themselves. These stakeholders collect and manage their data in different ways, making the data hard to find and use. Furthermore, personal medical information is very privacy sensitive. As a consequence, personal health data cannot be used easily by citizens themselves, by physicians, and by researchers. The Personal Health Train goes to the root of this problem by building FAIR data stations. FAIR protocols ensure that data are findable, accessible, interoperable, and reusable. These data stations are connected by tracks, which are strictly secured and protected. FAIR data trains are constantly monitored, and only trains with the appropriate validation may enter a particular station. To secure privacy, every data station has rules. Each...
No. I'm off the net. My password expired. I can reboot it, but rather than take time for that, let me summarize what you would have saw. So the Personal Health Train is an initiative there's a lot of interest in the Netherlands for this, and it's also, I think, now being recognized at a European level. It's recognizing patients' access to their own data and control over their own data. It's based on the FAIR data principles. And I think the other interesting thing about the Personal Health Train idea is that architecturally, it's a little bit different than the way we usually do analytics. So usually we have a data set, and we, if you want to analyze that data set, you transport the data, and you can run your analysis. Realizing that these data sets more and more are too big to move or too sensitive to move, like outside of a hospital firewall, the idea of the Personal Health Train is that there are algorithms that actually go to the data. They're smaller, they can move easier than all the data, and they can go to these different data stations and knock on the door. If they have the right access, they can dip in, do an analysis, and in many cases leave behind the sensitive parts and just take the results and go to the next station. That's the idea behind the Personal Health Train. There's a lot of interest in there. If you want to know more about the Personal Health Train, you can talk to me afterwards. But given what we just saw with these three examples, and then this last example that we have in our own group, consider the Personal Health Train scenario for this case. So HealthTap is receiving, I think by now they have something like 6 billion submitted questions and answers. So it's a huge data set. And from there, you can begin to see trends in concepts that are being asked in the same question. So you begin to see hypotheses implicitly emerging in that data set of 6 billion queries. When that happens, you could dispatch trains. In a world of FAIR data, you could dispatch trains to FAIR data stations like PubMed, like DICA, like Euritos, where these trains could then start looking for more evidence of these associations that are surfacing as hypotheses in the HealthTap case. So this is just an example of where there's lots of different data sources out there. A patient has manually gone out and with his own consent has been able to, he can have control over his own data. He's been able to piece together things about his own condition. But there's no reason why computers cannot do the same. I got a couple more case studies that I'll just run through, and maybe it helps to fill out in your mind what the capabilities of FAIR data are. This is an example from our own research group. So we took time at one point to verify what's called the Phantom 5 data set. This is a large consortium that did functional annotation on the mammalian genome. And in Phantom 5, the big question was, can we identify all the transcriptional start sites of the human on a cell type and tissue type basis? Because we want to know, if there's more than one transcriptional start site, is that a source of regulation for gene expression? So a big consortium looking at RNA-seq analysis from different mammals, humans, different tissue types, big data set. But we took some of that data, and I think it was 800 rows by thousands of, or 800 columns by thousands of rows. We took some of that data and we verified. And what we did is you see here the data model that we built for the data that we verified. And what you see is a chromosome location, and there's the beginning and the start position on this chromosome. It's chromosome number 18, and these are functional annotations coming from the Phantom 5 project, and they're linked to a particular gene. And there's a way you can write a code that allows the computer to understand that data model. So we're really just rewriting this tabular data in this format. But we can do the same thing for what we call the Leiden Open Variation Database. And this is a curated database from physicians at Leiden, or whoever has an installation of this data set, this database, where physicians that receive variant data from a patient can feel free to annotate phenotypes and notes about these variants. So some of these variants may be pathogenic, some of them might be uncertain, but at least you have physicians very carefully curating that data. But again, we reached in and modeled some of that data in a way that looks a little bit like the Phantom 5. We have chromosome position, and we have a kind of annotation, and it is a particular kind of variant. So we are now marking up the data in LOVD in a machine-readable way, just like we did the Phantom 5. And what this enables us to do then is launch a query over these combined data sets that allows us to ask a question that spans both. So we're getting interoperation now between these two data sets. And it's easy enough to show interoperation between any two data sets, but the scalability here that you're buying yourself is that now if there's any other data set out there that has unique identifiers and is built using existing ontologies, you can automatically link this out to these other data sets. So for example, if I have a particular gene, I can link out to all kinds of linked data resources that are related to that gene or to other genes related to it. And what we were able to show by doing this experiment was that 20% of the known disease-causing variants in LOVD were actually variants in the transcriptional start sites and not in the proteins themselves. So these are regulation-based diseases. So that's an example where we could show a new result by having verified these data and doing a simple query, but now this data is reusable for many other types of queries. Here's a third example. We also have a project in rare diseases, and here we have a colleague, and he's looking for biobank samples, in particular blood samples, for patients that have a particular phenotype, in this case a short neck. And so he has a very specific kind of interest here. And what we were, an interest that crosses over different repositories, different types of data sources. So in this case, biobanks and patient registries that keep track of patient phenotypes. And so what we did is, with the RD Connect project, we've been building linked data out of registries and biobank sample catalogs and pieced together a set of template queries that are asked in a very common way by people who are always working with biobank data. And we could formulate Claudio's query using one of these templates. And so this is what Claudio would see. So he has kind of drop-down menus that allows him to pose his questions. Behind the scenes, there's what's called a Sparkle query being formulated using that information that can go out and query over these linked data sets. And what he's pulling back now is, in fact, information about what samples are available, where are they. He knows that they're related to patients that also have short necks. None of these data are coming from a single database. They're coming from interoperable data sources. And I'm going to leave it at that. It's just a sampling of the kind of things that are possible when data is more interoperable and when it's intrinsically findable and has set accessibility specifications on it. So I'm going to reach into some bonus slides. These are things that we didn't see this morning, but we could have if we went all the way to the bottom. Okay, here we go. So I'm an academic researcher, I think very much like many of you. So I think you recognize the story as well. 1992, I'm a graduate student, I'm a PhD student at the University of California in Los Angeles. I got interested in RNA, chemistry, biochemistry, and RNA structure. And so I went out in different data sources by hand. I manually made a data set. And this became published, and it became, we analyzed it, got some nice results. And then it came time to graduate. And at this point in time, my data management plan consisted of downloading all the data from the hard drive on the computer we had in the lab onto 3.5-inch floppy disks that maybe some of you don't even know about. Each disk contained an amazing 1.44 megabytes of data. But I actually had a stack like this contained my entire PhD directory. All the papers I've written, all the data that I had. Nowadays, this data is securely stored at my mom and dad's house in Shelby Township, Michigan, in the basement, in a shoebox, and this is quite literally. I was able to find this over last Christmas. It was right before I came to here. And so these disks are probably not readable anymore, even if you had a drive that could do it. I'm not sure I could understand what's on them. So in principle, I found the data, but they're probably not accessible. They may not even interoperate on Excel anymore. I don't know. Moving on, 1997, I get a postdoc at MIT. And again, we do some research. We were able to eventually publish this in Science. In this case, I synthesized a bunch of RNA sequences, artificial RNA sequences, made mutations and was trying to study that. I analyzed the activity of these ribozymes using gel electrophoresis. And so this was a lot of work. There was hundreds of gels, and scanning them, quantifying them, normalizing them, eventually able to make sense out of the data. In this case, my data stewardship plan was backing everything up on what's called a zip drive. And this advancement in technology allowed an amazing 100 megabytes to be stored on a single disk. Today, I have no idea where those disks are. They're gone. And the hundreds of gels that I ran, they're gone. Sorry to say it, but that's just the way it is. And although this is kind of tragic, I know it's probably typical for many of you as well. This is just the kind of things that happen. But then I remembered, this was right around the time when supplemental data was starting to come online. So I went back to the Science article, and in this footnote, there was a link to the supplemental data. So, okay, great. When I put that link into the computer, Science, the publisher of that article, is giving me a 404. So, okay. What about a Google search? I got an amazing 9 results back from Google. None of them were my data. Okay? So then I write a letter, and this is now October of last year. So it's more than a year ago. I wrote a letter to the publishers at Science and explained the situation and asked them if there was some way we could recover my data. To this day, they have not replied. And then, and I forget even how this happened, but so this URL is ending in HTML. The footnote, the original footnote, was ending in SHL. So I forget how this happened, but somehow, if you just change it to HTML, you get my data back. So it's a hidden link, and it's there, but nobody knows how to find it. So this is just the example that I give of poor data stewardship, but also probably very typical data stewardship. And just that, this is a premier scientific publisher with broken links and these things. Yeah.
Are you surprised about that?
What's that?
Is that surprising?
No. In fact, there was a nice kind of small study that was published in Nature looking at basically the half-life of links on the web. And links degrade over time because it costs money and time and technology changes. So if you're not paying attention, if you're not putting in the proper data stewardship, links will just degrade over time. And I think it's after 20 years, 80% of the links are gone.